Good evening, everyone, or good morning, anywhere where we are. The goal of this call was to update on what we are doing and the time scale sometimes that we have in academic setup is different to what we would really want to be. So I want to make sure that I can keep everybody updated in what we're doing, where we are at. And we have the opportunity through the foundation to share what we're doing together in a 10 Zuckerberg meeting as a result of the Rare as One grant. Before going there, we're going to be sharing two posters. One is going to be from Sailor in my lab, and another one is going to be shared by me. Before doing that, I just wanted to make sure I could share with the foundation first what we're going to show. And it's basically a research in progress where we are at. And I do have great news, so that's actually wonderful. And what's the plan in terms of the bank for us and our sharing capabilities right now? So please do ask me any question. This is very whoops, sorry, I'm like starting the way around. I don't think it should be this way. Give me 1 second. That for some reason this got in the way. Okay, now we're talking. So the first thing I did want to share with everyone is what's the mission that we have in the lab. And basically, the overarching goal that we have is trying to advance knowledge in rare diseases with epigenetic origin. And that's why I started working in my postdoc, and then I had an amazing opportunity to work with the foundation. And it's impossible not to love the passion and to feel part of the KAT6 family by working with you. So I kind of took this into my research program, and we really want to make sure that we can create resources that will move forward what we know in KAT6A and KAT6B. And the way we're doing this is with a very strong emphasis in basic science, developmental biology, even though we believe that every kind of scientific discovery that we make is translational, even at any scale of the scientific knowledge. We believe that everything will be translated to benefit human health, in particularly disorders. So we do basic science. We are very proud of taking a patient focused approach. We want to hear where the problem is, and we want to make sure that we can ask the right questions at the right moment. We also guide ourselves with open source biology, so everything that we create is shared, from tools, resources, protocols, everything. And I also think it's very important to know that I'm super focused in mentoring every single trainee that goes through my lab in having this patient-focused center, because it's patient-centric focus. Because what's the point if we don't if we don't allow other young investigators and scientists to follow the same path that to make like a domino effect. But having said that, I shared this slide with you a while ago when we just started putting together the bank. I just brought it back again just to kind of see where we are at. We have two main objectives with the iPSC Bank from my lab. Beyond that, since the cells are going to be distributed to anyone who requests them, the capabilities of these are way broader from what I'm sharing here. So the first objective is establishing the bank. And I'm not talking about just reprogramming cells, but doing it with high quality standards. So we know that every single researcher who requests a line will request from every patient. We will have the parental control, the sex match control when available. And we'll do all the QC. We're doing all the QC that will accompany that cell line. And we are generating more than 3 clones per cell line. That means that from the same patient, there will be 3 different cell lines that they're supposedly to be the same, but it's a way to guarantee that the findings are reproducible. So that's what we were going to put in the bank. But in my lab, we have at least 10 clones for patients. So one thing is what is going to be available formally in the bank, in the core facility from the Center of Regenerative Medicine, and the other thing is what we also have in the lab that we are banking and keeping for the foundation and for researchers in general. So once we have the bank from we want to capture every single patient that is willing to, and family who's willing to contribute. And the goal is to honestly represent the whole diversity of our patient population. And from those lines, we also are going to generate what is called isogenic control. With isogenic, I won't get too technical, but the goal is to make sure that all what we observe, all that we study in a patient-derived cell is because of the KAT6 pathogenic variant, and not because there is any kind of genetic background also contributing to that, which is super important to understand as well. That's why collecting the most amount of patient samples is very important, because we know every single person is unique. But at the same time, scientifically, it's important to make sure that we can understand and dissect what's caused by the KAT6 variant and what's caused because of the patient background. So those are the two main goals there. And the second one, it's a goal that was born through the conference that the foundation organized earlier this year, and it's to try to understand what are the acetylation marks in these cells. So if we remember that KAT6 actually is a acetylates protein. Mostly in our case, we're focusing on the histone 3 acetylation marks, but it's also known that acetylates other proteins. So we want to understand how does that look like when we have these PBMCs are the nucleated cells that we isolate from the blood when we draw blood from patients. How are those acetylated marks in histone 3 behaving, or how do they look like in PBMCs? How do they look like when we reprogram with cells, when we make these stem-like cells from patients? Are they the same when we're differentiating them into a neuron, when we're differentiating them into a gut cell? So understanding that, I think, is going to be very valuable, not only for us in the lab from our interest in epigenetic disorders, but in general for all the community. And we from that meeting that we have, we outlined a way of doing it, and we took the advice of the expert in that meeting, and was super helpful in suggesting a high throughput method that now I'm super glad to say that we were able to optimize it for methylation marks, and now we're moving into acetylation marks. So I think that's good news.
Angie, can I just make sure I understand what you mean by marks? Do you mean biomarkers or what?
Oh, no, it's like KAT6A and KAT6B will add an acetyl group into the histone. Also, it's known to do in different in other proteins like p53. So these marks are it's known that it could acetylate 3 different amino acids in the histones. We want to assess in these cell types, is there any fingerprint, any signature specific of KAT6 in KAT6A?
Fingerprint. Okay.
Exactly. And these, I won't say that could be like a biomarker, or that we don't know enough yet, but there is a huge interest in understanding all the marks, not only the ones that have been published that are actually known to be caused by KAT6 and KAT6B, but we want to just do a whole profiling to see whether there is another mark coming up in another amino acid and another lysine because of the variant of the patient. So we just want to learn. It's very, very basic science, but I think it's going to be very helpful for the whole patient community and downstream project.
Thank you. Okay.
Thank you, Andrew. That's actually a good question. Sorry, I didn't clarify. This is an overview that I've also shared before. And this is like the big picture. What does it mean to make, to establish a bank? And generally, different labs around the world will create cells. When the cells are published, one may or may not decide to share them. And that sparse effort. Sometimes the quality controls are not the same. Now we are saying, okay, let's just put everything together in one place, let's standardize the highest quality controls that we can have, and let's share them. Let's make it open. So that starts with, as always, thanks to the help and the support and the contribution of all the families, patients, and the foundation. So we try to capture all the patients that want to donate blood. We always emphasize that we do need the genetic report, because we are not doing the diagnosis. Sorry, hi.
That's
I don't know if there was a question.
what I need. You're smiling.
Please do let me know if there is any question. So we capture most of the patients that we can. We have received donations of patients that did not share the genetic report. Is very important for us in order to be able to reprogram those cells to have the genetic report, because if not, we cannot move forward. So I'll ask Sailor to contact those patients again, so we can have the genetic report from all of them, because we are not reprogramming cells that do not have that report for just to preserve resources. So once we have those, we isolate specific cells from the blood. We do the reprogramming. I'm going to talk a little bit more about that now, because we're planning now the second round of reprogramming. I want to share with you where we are at. And once the cells have been reprogrammed and have passed quality controls that we do in the center, then they are shared in a catalog, online catalog, so anybody can just go in there, say, okay, these are the cells, and contact directly the CReM bank. And if it's inside the foundation and the research is started in the foundation, and there is any other pressing need of information, they can just contact me directly, because we can speed up a few steps that are a routine in the bank. And the bank, it receives requests from all around the world. So I can support to speed up a few requests if that's necessary. And then the wonderful thing about these iPSCs that are induced pluripotent stem cells is that we have, from the patient, we make those cells that were blood cells. We say, forget everything you know about being a blood cell, and become a stem cell-like a line. And with those, then we can instruct them to be heart cells, to be brain cells, lung cells, gut cells. So there are loads of possibilities. In our lab, we work with established protocols, but all my neighbors, all my colleagues are pioneering protocols to generate from these specific stem cells unique or very distinctive cell type from lungs or from gut or heart. So I think that we're in a good place to actually not only say what we're going to do, but actually do it. And with this, we can then start talking about more precision medicine or testing, and of course, what we do, which is basic science.
Actually, on the precision medicine, Angie, because you say they're functional testing, drugs work for all. Couldn't it be that you find out some that are working for some of the patients, in a personalized medicine way?
That's a good question, Andrew. And I don't have a straight answer for that. The way we are approaching this, when we say, okay, which lines, sadly, we cannot reprogram all the cell lines at the same time. So we say, what are the lines that we're going to start reprogramming first, even though the objective is to reprogram all of them. We try to focus on cell lines or in samples in which the patients, or in which what it's observed, the clinical manifestations are shared for most of the patients. So everything, all the discoveries that can be, or the advances that are done in those particular samples or from those patients, can be somehow translated or extended to most of the patients. That's not how it always works. It's very hard. Yeah. And the way, that's why our goal is to do it from every single patient, because we have rich genetic information from where the pathogenic variant is located, from the genomic background. It's only just one of pathogenic gene that has been identified. Is there any other component? All that is knowledge that we want to represent in the bank. But yes, you're right. We don't know. The only way to know this is to see. I don't think that one drug will help everybody in every
Exactly.
single
Me neither. Yeah.
So let's say I would love for us to say one day, this is the driver of most of the clinical presentation of patients. So if we can understand this driver, and with this driver, I don't know, I might be talking about neurons and muscle connection, I may be talking about mitochondria, I may be talking about whatever in the scientific community we can discover, then it's a bit, we're closer to say, well, if we tackle this main driver of the clinical presentations, we are going to be in a better place to say, okay, this might benefit most patients. But we have work to do, and that's why I'm like full speed.
Yeah, I mean, that's the ideal, that it would, they'd be functional for drugs that work for all. That is obviously the ideal.
Yes,
yeah.
Exactly. We start with the ideal, and we'll work from there. You're absolutely right. So this A part, which is collecting the blood, everybody was extremely helpful in the foundation, just helping and supporting us to make this happen. And now this is one of the things that Sailor is going to share in the conference. Here we have the KAT6A and KAT6B cartoon. This cartoon is not the ones that you're going to see in generally in the publications, because we do it from scratch. We have a methodology for making this so we can map accurately every pathogenic variant with the newest annotation, and we do it on scale. I'm happy to talk about this in a different talk, but I just want to make sure that people know why these cartoons might not be the exact same as the ones that are reflected in the publications. And here she just put a few of the variants that we have represented in our bank, that have the genetic report. And the wonderful thing about this is that we are trying to collect. We're not making priorities on the samples that we collect. We really want to capture every single patient that if we can. And I'll keep doing this until the university is saying, okay, Angie, that's it. But we will work very hard to make this happen by next year. And the reality is that we do not have enough KAT6B patients. So right now, 75% of the samples we have are KAT6A, 25% are KAT6B. And it's important for us to also represent a male-female population, because especially when we're talking about mitochondrial disorders, we want to make sure that we're going to account for sex as a biological variable, and we want to consider that. So we are making sure that we have both controls, male and female, from both patients. And in the lab, when we're talking about engineering these in isogenic lines, like grabbing a cell that is well standardized, well characterized, and introducing the pathogenic variant and correcting it, introducing it and then correcting it, we are going to do this in females and in male as well, just to make sure that we can cover all the bases, and that every discovery that we do is as steady and strong as we can. So what do I mean when I say reprogramming? So Sailor did an amazing job at depicting this in a cartoon. It's long. For me, it's super important, because the times for us, the urgency is here, is present, and I am super aware of this. At the same time that I'm realistic with the timing. So Sailor started doing this fresh out of college. She joined the lab. She trained on everything, and one of the things she trained on was in reprogramming this cell, which is not. I was not training reprogramming cells, so we acquired this expertise in the center, and now she's successfully doing it. And the whole process takes, in the best case, between 2 and 3 months. And it was very important for me to transmit this, because when I say, well, yes, we'll reprogram, what does it mean in terms of timing? For us, in our hands right now, and at the capacity that we have, and with a person who her main project assigned is a KAT6 foundation project, but she's also doing many other things. And in her hands, it took her 3 months to reprogram 2 cell lines. She started with 4, and she could successfully reprogram 2 cell lines. And I also want to discuss this because sadly, we are at the moment where the efficiency for reprogramming the cell from day -9 is when we are making the cells that we isolated from blood. We need to expand them. So that's the minus part in this first part of the timeline. And then once she does that, she needs to generate loads of different colonies and clones from the same patient and expand. So she's handling loads of plates, and the efficiency, not every single cell or every single colony is appropriate. It's not easy to reprogram efficiently all of them, but we only need 1. So we save everything, all the samples along the way, and we'll try again for those that we couldn't reprogram again. So from a process that takes 2 to 3 months, she started with 4 lines because it was the first time she did it. She could reprogram 4. For me, those are good numbers. And now the second round of reprogramming, we're going to start with 8 lines. And we are training 2 additional people in my lab to also support in this enterprise with the goal of reprogramming most of the, if not all, the lines, all the patients' lines for sure, but we're also talking about the sex match parent control for next year. So that's our goal. And this is what the whole process looks like, load of taking care of the cells. And a very important part of this whole process is over here. Once we say, okay, we made it, we have these cells that are stem cell-like cells, we want to make sure that they can actually be directed to be any other cell type in the body. That's what we call pluripotency test. Are they actually pluripotent? Can we make, do they have the ability to make brain, gut, heart, muscle cells? So once they pass that test, which is a test that we do in-house, the next step is to send them to a company to make sure that through all this process, we did not introduce any chromosomic abnormality, because we are really forcing those cells to forget everything they know about what they are and then to become embryo-like cell. And that sometimes can introduce chromosomic abnormalities, and that's this karyotyping step over here. And that's exactly where we are at. So this Monday, we sent all the clones that we generated from these lines for karyotyping into one of the companies that we think is going to be giving us the results in 2 to 3 weeks. So with this, I wanted to share what do I mean with karyotyping. These are the 2 cell lines that she successfully reprogrammed. Here you will see A and C are the 2 different cell lines that have a green fluorescent. That green fluorescent is showing a mark for a protein that is telling us these cells are pluripotent. And next to it, you have, this is how IPS looks like. And if some of you went to the lab during the conference, you will remember this, because we did show them, we had them there. And that's one of these. This is one of the pluripotent tests that Sailor made. We have 3 clones. We're not banking 1 clone, we're banking 3 formally in the bank, and we have in our lab, just because we want to make sure we are not wasting all the effort of these cells. We're banking every single clone that looks good. We're banking it, and we are right now karyotyping and doing all the QC to 3 of those. The next reprogramming is going to start at the end of September, where we come back from this meeting, and we're going to ramp up now that we feel comfortable that we can do this successfully, we will ramp up our reprogramming pipeline, and we are training 2 additional trainees in the lab. I think it's very important that we can have more people helping with this, because it can we can be doing this first. So we're trying to make it as fast as possible. And I do hope we can have all the samples that we collected from the patient donations in 2022 and in 2023, that we can have them reprogrammed by 2024. I do want to say early 2024, but I've learned that my times are not sometimes the times of the cells, so I'm trying to be cautious here. And the most important thing, and I don't, we talk about, okay, let's reprogram, let's do this, it's a bank, it's open biology. I think it's very important to understand what this means in terms of price for the scientific community. Right now, the Center of Regenerative Medicine offers this as a service. If you're a researcher and you want to reprogram cells, you have patients, all that cost is reflected in the webpage. You can just go there and see it. For the basic reprogramming, 3 clones, 2 vials, just the reprogramming is $7,500, plus all of these other things like the karyotyping test. But we don't do DNA fingerprint, but karyotyping, the mycoplasma test, the PVNC, all of those things, we are doing them in-house, and we couldn't do this without the support of the foundation and without the support of the families. And it's a collaborative effort between the foundation, our lab. We're dedicating time, resources, and the CReM is giving us the space to breathe and to do this and the training. And I do want to emphasize this. I honestly think it's a great resource for the community, and I'm not beyond the research of our lab. We're doing everything we can to make sure that we can gather actual knowledge and actually push discovery beyond that, beyond of what we want to do. I think it's a great resource for the whole community. And I do want to thank all the lab, because I think that you met all of them. Now my lab has closed the door to hire anybody else. This is the lab until we get more fundings, and they're all super excited about working with the foundation, and they're committed as much as I am with the patient-centric focus that we have. And most of this is thanks to the openness and the trust of the patients and the foundation. So I do want to thank them and the